We're next going to hear from Professor Morten Meldal, Nobel laureate in chemistry and head of the Center of Evolution and Chemical Biology, University of Copenhagen. While it is rare that new scientific findings occur in absolute isolation, the ability to recognize and realize new targets requires a well-honed scientific mind. In other words, it's not just the science behind a new idea, but the understanding of the potential of the idea that constitutes great science. The concept of click chemistry matured simultaneously in different laboratories around the world in the 1990s. There was an urgent need for quantitative chemical reactions to cope with the pressure from combinatorial science to synthesize, screen, and identify millions of compounds. Professor Meldal and team discovered the click reaction in 2001. The mechanism of this reaction will be discussed and its applications will be presented, as well as we'll consider the importance of serendipity. Please welcome Professor Meldal, who will speak on Molecular Click Adventures, a leap from the shoulders of giants. Welcome.
Thank you to Laurie, thank you to Bengt, and thank you to the organizers for this opportunity to talk to you today. I will lean a lot onto the shoulders of the four giants that we have heard speaking today, because there's a lot of things that we all have in common, and I think there's a lot of the ideas that will sort of resonate between the different speakers here today. We are all direct descendants of Homo egoistica as Nobel laureates. But I would also like to say, as a young researcher, don't think about the Nobel Prize, because then you're never going to get it. So we have just heard that chemistry is everything. This is one of my mantra. Chemistry is everything, and if chemistry is everything, and it really is, when you feel pain, when you feel sleepy, when you're hungry, it's all chemistry. Chemical compounds that interact in a very complex manner to elucidate these feelings, or give you these feelings and needs. And if that is really the case, if chemistry is everything, and we are modifying this world at an unprecedented rate by chemistry, if that is really true, then we need to teach not only the scientists, but everybody about chemistry. Just like we learn to read and write, everybody needs to know what this world is composed from. And that could have enormous effects in the way that these challenges that we're facing today, both politically and environmentally, can be handled. So I also agree totally with the former speakers that we need to do research without constraints. So allow the serendipity that comes from the discoveries, but under some sort of responsibility. And then our funding systems need to change. That's very, very important for you as young people, and you should advocate for this. Because when we make all these applications for funding, we are applying these to wordings from the funding agencies and not to our own thoughts about what is important. So I shared the Nobel Prize with Caroline and Barry, and this was a great pleasure to be up there. Once I finished my study, I got to the Carlsberg laboratory. This is the Max Planck or similar institution equivalent in Denmark. And we started out by looking at what is really needed, what is needed in order to investigate the world is a lot of molecules. And for that, I introduced these kind of polymers, which are very unique in that they are completely compatible from toluene and all the way to enzyme buffers, which means that we can do everything we want with these polymers. We can synthesize the molecules, we can analyze the molecules. They are completely transparent, so we can use light sources to investigate our structures that sits on these molecules. And we're not making horrible mixtures. We are making clean molecules, one on each little bead that we produce of these polymers. And you can see we have done quite a large variety of different reactions on these solid supports and taking them into quite complex scenarios in proteins and peptides. And we have also investigated enzymes inside these polymers. So we can actually now combine polymer chemistry, organic chemistry, and enzymes. We can even immobilize human or yeast cells in these polymers and do chemistry with them. And we can produce these polymers quite in a regular fashion. And I would like to just show you how we do that, because I think that is a very nice process in which we make a continuous flow synthesis of a single bead at a time at a very high rate. So we are using a pump and a silicone oil solution that is going through this Teflon coil where you have the polymerization, and then we just collect the beads. This allows us to do beads which are quite unique in that they vary very little in size. You can see how regular these beads are, and that means that we can actually do quantitative measurements on these beads. So we have a whole platform technology that we have developed in order to study chemistry from an evolutionary standpoint by doing combinatorial chemistry. We start out with a combinatorial process in which we use each bead as a separate container for chemistry. So it's your reaction flask, the round bottom flask, and you can see we have a lot of them. We can then find active beads out of a library, one compound on each bead, different compounds. And these are the active ones. We can sort them out, find the molecule, and inject it into a mouse and see directly, here's an effect. We can synthesize all sorts of things on these beads. So it's not only peptide chemistry that we can perform. Here we, for example, grow cells on the surface of beads, and they act as detectors for activity of these molecules that are associated with the beads. So here is some inactives and one active bead that we take out and analyze. And we have now developed also a way to analyze these beads that is like a barcode when you go to the supermarket. You take the thing from the shelf and you just read the barcode and you know everything, the history of this commodity. And this is the same with our beads. We have this kind of matrix that we can use to read the structure directly and at the same time read the fluorescence signal as an activity on this bead. So we can actually speed up discovery quite dramatically in this platform. This is how it works with the combinatorial chemistry. You probably know this, but we are dividing the beads up. We are coupling different building blocks onto the beads. We mix them up and we repeat that many times. But on each bead, we just have one kind of molecule. And we can then test these molecules by adding an enzyme, for example. So in this whole process, one of the reactions that we discovered was the Keurig reaction. And it doesn't look very impressive, but it has had a major effect on the way that people do chemistry in materials, in biology. Everywhere that you use chemistry, you can use this reaction orthogonally to all the other chemistries that are there. So we have a huge toolbox of organic chemistry that we are using for all sorts of synthesis. And then at the end, we can use this reaction to combine different functionalities into one molecule. So this is used in a lot of different ways and was presented by Christian Tornø, who was the PhD in my group at the time, in the APS in 2001. So what is this reaction? Yeah, here's a complex chemistry that you mechanism where you have the copper atoms associating with the alkyne, you have the substitution with the nitrogen, with the A side, and then you get the cycloaddition, loses the copper, and you have this cycle. And there are crystal structures that demonstrate all of these intermediates, and we have a very clear evidence for the two copper atoms in very super active catalyst where you pre-organize the copper atoms with the right distance. So we know a lot about this reaction, but let me just explain that a little bit more visually. So here we have an alkyne. An alkyne is a sack of electrons. It's high electron density, and it looks out for positive charge. So here comes a copper 1 plus. It attaches to the alkyne and acidifies the proton that sits here. So the copper takes the place of the proton and forms this one, which is not very reactive. But a second copper atom can attach itself to this one, and then it peels off the electrons from the alkyne to give a very reactive molecule here. This is the empty orbitals, or the LUMO, of this molecule. And then we have an A side here, which fortunately fits exactly both with the phase and with the geometry to overlap here and give the electron density to form new bonds. So what happens is that first you form this metallocycle that loses one copper atom, then it loses a second copper atom, and then we have this new connection that is just like a glue. We can glue molecules together using this reaction. So here we are, 3 proteins. One is a protein that can kill cells. We have developed that. It interacts with an inhibitor of apoptosis. And the other one is recognizing a cancer cell. And the third one is a cell-penetrating peptide that we have made that is very, very efficient. So if we now combine these 3 using different technologies, in either synthesis or amber stop codon technology, we can then combine these 3 into a container with copper 1, just steer it overnight, and then we take out the molecule, and it's almost pure. We can now use this molecule in the reaction with a cancer cell. It recognizes it. The cell-penetrating peptide takes the other component into the cell, and then the cell dies because of the apoptosis. So we can use this in ligation. We can modify surfaces, we can modify other proteins, we can do robot molecules. So this is the facilitation of making a molecular robot that is multifunctional. We can do structural stabilization, just like disulfide bonds in proteins. We can actually stabilize a particular structure using this chemistry. Surface immobilization, we can make new polymers, affinity profiling, and so on. There is so many different applications of this orthogonal chemistry. So the only caveat with click reactions is that we have a redox issue, because copper 1 is not very stable. It very readily undergoes oxidation with oxygen to copper 2, which is completely inactive. You can see the shape and sizes. The size is different, and it doesn't interact very well with the alkynes. So here we have a method that Sharpless developed of adding a reductant that brings it back, so we always have copper 1. That is quite nice, but it's also a great danger, and that is a danger because we are forming these reactive oxygen species. And while that is fine with small inert molecules, it's complete disaster with proteins. And you can see that here, where we have synthesized a protein of 14,000 Dalton. And when we do this in the presence of air, we get all these oxidized materials. So we have to avoid oxygen, but other than that, it's a perfect reaction. Here's an example that we have where we have used click chemistry. This is the Japanese horseshoe crab, a fantastic creature. It has no humoral immune response. It lives entirely on its innate immune system, which it has a blue blood that we use for medicinal uses with 2 macromolecules that encage foreign organisms, and then it injects these very toxic compounds in there. So we were interested in these cannonballs. And if you look here, this is a structure. It's a beta hairpin with 2 disulfide bonds. And we would just like to see what happens if we now click instead of using these disulfide bonds. First of all, we found out that it was actually this kind of cannonball. It forms a dimer by NMR spectroscopy. And secondly, these dimers, what they do is they find a negatively charged cell surface. They are all positively charged. They unfold and form a pore, the pore in the cell membrane, and everything that is inside the cells come out, and the cell dies. So this is how these works. And the question is now, does it work as well with a click as it does with disulfide bonds? And we tried out coli, staphylococcus, salmonella, and bacillus subtilis. And as you can see here, the natural one, which is TP1 here, is very active for the 3 first one, but in the 4th case, it's completely inactive. Most likely because the disulfide bonds are reduced in the process. So here is the clicked compounds, and you can see they are really, really quite active all the way. So click is good for antimicrobial peptides. Here's another example where we use click chemistry for structural purposes, or to link 2 things together. Because this is a protease, the protease, this is a virus we saw earlier on. And this virus actually has a protease, and therefore, if we express it in coli, we have the problem that it's toxic because the proteolysis kills the endogenous proteins in the host cell. So the coli bacteria are quite unhappy with this expression. You get a little bit of the protease, and then everything dies away. So we thought maybe if we cut this in 2, there's an opportunity to express these separately, the 2 halves, and then recombine them. We have a little trick because we insert a click residue here and here, and then we use the click reaction to combine them later on. And now coli is very happy. So here it is with the stop codon, the AMPA stop codon suppression technology. We can insert these 2 residues here and here. And when we combine the 2 and use a copper 1 source, we can make a full protease. And now the question is this as active as the natural one? And in fact, it's not only as active as the natural one, it's a little bit more active than the natural one. So we are very well off. If we show this a little bit more scientifically, here we have the 2 loops that we are making with a click reaction. And we tried both of these, so either 1, 2 or 3, 4 in this position. And as you can see, the short bridge did not succeed completely. We get some of the click material, but not all of it. And here we have a complete conversion, 1-to-1 into 1 new protein. So this is what you see up here, the 2 sequences here combined. I have taken the liberty of putting in a few slides from the Nobel ceremony. And this here is the most intimidating moment in my life. I gave a lecture for probably millions of people. I made it through the lecture, and so did Barry and Carolyn, and here we are enjoying the moment after the lectures. So The next thing I would like to demonstrate is a little bit philosophical, because there is some sort of economy in the way that nature has constructed things. So the 20 amino acids is by no way optimal for structural purposes, but is optimal for the entire machinery of producing proteins. And therefore, we as chemists can actually benefit from the 5,000 amino acids, alpha amino acids that are available. We don't have to think in the 20 natural amino acids. Because if you think about the natural amino acids, you have tacticity. All of them are the same configuration at the alpha carbon. And we know from polymer chemistry, this creates huge secondary structures from polymer chemistry. And here we have a protein, papain, which is full of alpha helices and beta sheets inside. It's a huge protein for a very small function here in a small active site. Why is that? But that is simply the economy of protein expression. So life has chosen to take this extra burden of making large proteins to be able to pack secondary structures together to form a hydrophobic core, a hydrophilic surface, a cavity that matches the substrate and the active site residues, in addition to a mechanistic property of the protein. But if we just took some L and D amino acids instead and combined these, maybe we could make much smaller proteins that would be synthetically accessible and would allow us to use all the 5,000 amino acids. So here is a way that we do that. We take an unfolded thing, we fold it up in whatever conformation we can get from that. We establish a hydrophobic core, and we stabilize the protein with this hydrophobic core. So this is what we call mutational molecular dynamics. We calculate, we mutate, calculate, mutate, calculate, mutate for a whole week, and we have a fully functional protein that folds around a hydrophobic core and has a hydrophilic surface. Completely artificial new protein. These are some examples of small proteins, but we made quite a lot, and this is how we do it. We start with a random DL sequence from alanines. We make a fold, and then we start substituting in and taking care of all of these things, electrostatics, hydrogen bonds, and so on. And when we are happy here, we go down here, introduce some click bridges and catalytic residues, and we synthesize and measure activity. So this is a way to make enzymes, and here is one of many projects we have done on this, where we synthesize serine proteases. You can see here's an aspartic acid, a histidine, a serine. Here's aspartic acid, a histidine, and a serine. And actually, this is a backbone carbonyl. But the fact is that we can make these very stable folds. So this is without any bridges that we get this sort of extremely stable folding. This is a long 10 nanosecond calculation, and you can see there's no changes in the fold. After we finish, we then establish the structure further with bridges, and then we design in proper residues that can do the catalytic reactions. And you can see here the MSD and the distances between residues shows very good stability over time of these proteins. We can synthesize them very purely, and here is a MS of the crude product that shows that we really have this protein. So now we make a combinatorial library using the combinatorial synthesis with a fluorescent probe here and a contact that can shut off the fluorescence. So if we cleave this substrate with an enzyme, we'll see a blue color, just like this. And as a positive, we are here using subtilisin BNP, which is it's a washing powder enzyme, but it's one of the most active proteases out there. And it looks like that. Here we are with the beads. They are dark initially, dark, and then we start getting a ring after 1 hour. Ring develops, and after 24 hours, there's a lot of actives and a few inactives. So the same study using our protease here shows that after 2 hours, we start seeing a little bit of blue here. It's not rings because this is now very small, so it diffuses very rapidly into the beads. Here it's starting to get more intensity, more and more. Here it's 24 hours, but you can see we have a lot of selectivity. Only these two in a large variety of different substrates are really active. This is 7 days, still selectivity, and this is after 3 months, then we start seeing cleavage all over. But we can actually synthesize a very specific protease in this way. We are now looking into glycotransferase and other kind of proteases. And enzymes are the main tools of nature to reshape molecules. So this is a very important entry into new ways of doing things. For example, can we make an artificial protease against a pathogenic protein on a virus? If we can, maybe we can use enzymes as a drug. These are metabolically quite stable molecules because of the D amino acids. So maybe we can make new kind of drugs that are not inhibitors, as most drugs are, but are actually actively combating a disease. So Mads, Sara, Rusen, Lina, and David worked on this. And here is the protease, and you can see the bridges that we introduced, the active site residues that we introduced, and the hydrophobic core surrounded by hydrophilic moieties. These are fully soluble proteins, 5 milligrams per milliliter easily, and they don't precipitate. So it's a really, really nice procedure. And you can see, if you have any knowledge about proteins, this doesn't really look like a protein. This is what it's looking like. Molecular recognition. We heard about molecular recognition. There are two things in nature that are really, really important. One is molecular recognition. That precedes everything that occurs in assembly, in processing. Everything that occurs starts with a molecular recognition. And the other thing is, of course, the processing itself. So here we are with molecular recognition. This is an antibody, the best nature's recognition molecule. It recognized almost anything, and it's combinatorially made in the body. You choose genes to combine to get you the recognition out here. We do combinatorial methods here for developing a beta hairpin that can recognize a protein surface. And we can do this recognition quite well with the modern technology of calculation. The way we do computation, it's quite easy to do this dynamic molecular dynamics or mutational molecular dynamics to get a very good recognition of the surface where we match positive with negative over the entire surface and get very good recognition. Here is 2 molecules, one here and one here. You can see this is attached to a bead and this is in solution, and I'll show you that just in a short while. It looks like that. We have the click bridge to make structure stable. We have a turn, and then we have structure residues on one side and combinatorial residues that can be changed on the other side. So here are 2 of those kind of molecules that recognize each other very well. It's like 700 nanomolar. It's not extremely good recognition, but on the other hand, I think it's the first time you have these kind of small molecules that are designed to recognize each other in this way. But we can also recognize proteins. Here is screen fluorescent proteins, and this is an interleukin, one of our immune proteins that is labeled with a fluorophore. And you can see here that there's a large difference between the structure of these two, and this is also reflected. Here we have a bright field image where you see the two beads. And when we then look in fluorescence, we only see the green fluorescence on this one, not on this one, when we add 60 nanomolar of GFP. Here we have the interleukin-1 beta added, and you can see only this bead lights up. There's no binding here. Very selective molecules. So we can now design binding for the first time here. When we do the clicking, we increase, these are non-clicked and these are clicked, and you can see the binding increase dramatically upon the clicking. We can also have two very similar folded protein like interleukin-1 alpha, interleukin-1 beta, and get only recognition of one of them. So it's almost like an antibody, and here's a very nice, this is what I showed on the first slide of these, beta body here sitting on the protein linked to the bead, another one with a fluorescence label. When we mix this and that together, you see nothing. Then you add the green and orange protein here, and you get this result because it binds to the bead, and then it recruits the other one from the solution at 20 nanomolar. Very nice recognition. We are back in Stockholm, and this is the day of the ceremony. You get the medal for a short while. You're not allowed to keep your medal. I was a bit surprised. I have to give it back. And then you get it on the last day. The reason for that is that Nobel laureates are usually like 40 years away from their discoveries, and something might have gone off in the meantime. So some of them have forgotten their medals in the ceremonial hall. Well, talking about that, this is about Alzheimer peptides. Alzheimer peptides have a huge variety of crystal structures here. This one was isolated from actual plaques in the brain. Alzheimer is a very important societal disease that increases with the increasing age and increasing sort of bad food habits in our society. So what it is these kind of fibrils that form in the brain. This is an electron microscopy image from Gramer. And we would like to look into that and see, can we do something with it? Can we, for example, take this fibril and cut it in the interface and then make molecules that bind to the interface in order to avoid this kind of fibrillation? And here's the result. We have made these two molecules, and this is the fibrillation curve without molecules, and this is the fibrillation curves at 37 degrees and at 25 degrees when we add the molecules. So we have something that works. However, the molecules are not perfect yet. This is the very best result we had there. And also, they are not totally soluble, so they're not good for injection, for example. But we can also take these peptides and add them to already formed fibrils. This is after, you can see this is a long time. So this fibril is quite stable, and then we add our molecules and we reduce the amount of fibrils very significantly. But there's still something that's missing. We are not getting to the bottom here. So my PhD student, Yuan Sang, did a new kind of molecule where he take this glucosaminic acid. We're making all of these beta bodies with D-amino acids for a particular reason. But these actually have exactly the same configuration as D-threonine, but they do have 3 extra hydroxyl groups on each one of these side chains. And he converted it into a very nice building block for solid phase synthesis. I won't go into details with that because it took him half a year to get there, but it succeeded, and he used them in the synthesis where you can see these GAs are this residue here. And we synthesized 5 different beta bodies, these beta hairpin molecules, and we matched them up to the 2 growth phases of the fibril. And then we tried them out. And these 2 here, 3 and 5, I can say now is the ones that are really, really efficient. Here you see the fibrillation with these 2 molecules for a beta 1 to 42 for which they were designed. Absolutely nothing. And this is the fibrillation without. Here you have the fibrillation of 1 to 40, much more soluble peptide, but also maybe not structurally as coherent as this one. So what we imagine happens here is that these extra hydroxyl groups takes the water, which is a very important part of molecular recognition. Molecular recognition is not about the 2 interacting surfaces. It's about the entire environment in the water solution. And it takes these shells of water molecules and bring them in together with that and make fragments soluble. So here is a fibril, and these are the sort of fragments that are taken off. And then when we sleep at night, we go into the REM sleep when the brain squeezes, and then we can squeeze out all of these fragments and reduce the amount of fibrils in the brain. That is a dream. So far, we have only made it in vitro in our plates. I had another postdoc, Martin Wolfram, who worked on metalloenzymes. So these, I won't go into details with that, but these are peptides which has carbines incorporated into the peptide that coordinates with the metals. And we had seen cleavage with metals before, so we thought maybe we can take an Alzheimer peptide and cleave it with these molecules. So he synthesized the substrates and the peptides and tried to cleave them. And it worked fantastically. He reduced the amount of catalyst, and it still worked. And he got down to nanomolar concentrations of the catalyst, and it still worked. That was quite amazing. But then he took it out entirely, and it still worked. So what is happening is something along the lines of what we just heard in the last lecture by Jean-Marie, is that we have a autocatalytic process where the peptides, the Alzheimer peptides, cleave themselves. And I think this is quite important. The thing is, we don't know if a beta is garbage or whether it actually has biological functions. Because the Alzheimer precursor protein is cleaved in many different places, and all of the fragments have different functions of neuronal growth and so on. So here is a starting material. You can see this is the first fragment, this is the second fragment, this is the third fragment. It's almost quantitatively going in one way. It has a kinetic profile which looks like this. You can see this is hours, so a long time, and then suddenly it happens. So what happens here is that you probably generate the catalytic fragments from very small amounts in the beginning to more and more, and then you get this kind of self-organization we just heard of into a proteolytically active substance. And this is another place where we overlay with earlier discussions of a combinatorial system like the RNA we heard from Jack about the RNA that organize. Maybe our proteases here is the same kind of situation where you have a lot of different things and you have the selection that organize into a catalytically active protein. So here is a beta 1 to 40. Nothing happens in the first couple of weeks if we don't seed it. If we seed it, we get immediate cleavage into many. So if we take a little bit of this active substance here, just a drop, and add it to this solution, we get complete cleavage. Yeah, and here is something that is mechanistically explaining what happens in the fibril formation. So the first thing that happens is that we form these donuts. These donuts are formed by 3 different segments that comes together, and at some point they form these kind of collapses into small sticks, and these sticks then organize into long fibrils. So I don't think that there's another one like this out there yet. So I think this is quite interesting. If we do the treatment, we don't see many fibrils, we only see some donuts left. Yeah? Okay, so here is what we imagine. We have these 3 fragments that can collapse, and this is on the bottom a 3-dimensional AFM of these donuts. Okay, that you have to take care of. We are back in Stockholm here, and this is my wife. She's a very proactive woman. So this is one of the 6 dresses that was at the Nobel Museum. She just called the Academy of Arts in Stockholm and said, "I want this at the ceremony." And they said, "Yes." So she got it, and when you dress up your wife like that, what happens is that she was taken over by Prince Philip and brought to the ceremonial dinner, and I was sitting down there in the back somewhere. She was sitting between Daniel and Philip, actually. But it didn't really matter, because the next day we went to the Royal Palace, and I got the primary spot. So about use and chemistry. Chemistry is everything. It's a very important thing for all of us, because that is where we are going to find the solutions to many, many different crises today. So I imagine we make videos and VRs like that for all the grades in the school. We just use 5 minutes in first grade and show them how 2 hydrogen atoms comes together and like each other and share their electron clouds, how they burn with oxygen, the very aggressive oxygen twins. And we can also use material from the internet, but this is not coherent. So I really want to produce that. And then you just call this artificial intelligence sitting here, and he says, "How can I help?" And she immediately delivers whatever you need at that grade, that class. I think this could be not only something good for Denmark, but for the entire world, if we can make that available. And it should be made not by teachers in the school, it should be made by hardcore chemists, quantum chemists, a few animators, and a didactic person who knows how kids actually consume this. Because kids, and you are not kids anymore, but kids like first graders have an amazing ability to store images. They see something and they remember it for the rest of their life. And if you think back on a happy event in your childhood, you see it very clearly. You can even see details of where you were and what happened. So I think this could be a very important access to the brains of young people and stimulate them towards understanding existence in terms of chemistry. Because chemistry is everything. And we need to teach the students that. We also need to understand that all discoveries, all great discoveries, are serendipitous, and that is because if it was obvious, it would already have been done. It is an observation that we don't expect that leads to the next discovery. So we have to be completely open, not only as individuals, but as a society, to make these serendipitous, groundbreaking discoveries that we have heard so much about today. We want productivity, quality, creativity, responsibility, and translation, and I think the scientific community is matured. It can do this. It don't need politicians to tell us what to do. And also, the funding has to be changed. No applications, funding based on performance, and not from a big pool like that, but from two different pools that take care of very different functions. One is to take the knowledge we already have and put it into application. The other one is to do fundamental, groundbreaking discoveries that will be the rescue of tomorrow. So if you're a young person, you have to select what you want to do. And I think that as a young person, you might change attitude a little bit towards trying to solve the problems that are going to affect you tomorrow. So here is sustainable materials, new production processes, efficient catalysts for energy, new degradable materials, solar technology, transition to hydrogen. Hydrogen is much better than electricity. I don't understand why we have to go through this period of electric cars. And new food sources. Find new food to feed all these people that don't have food today. Textiles. Textiles are damaging our world. And transportation. And here I've just shown Novo Nordisk new wonder drug because they're making such a buckload of money that they could pay for some of all this. And they are, because there's a new great initiative on way with Novo Nordisk Foundation where they are looking at CO2 sequestering. So if you are applying there, maybe you can work in one of these areas. And with that, I would like to thank all the wonderful people that I've shared my time with. You know, I used to say to my students, old people like us, we are like vampires feeding off your energy. And that's true, actually. So I've been feeding off of all of these wonderful people in the lab, and they have done fantastic things. And I also want to thank my family for being patient. I spent far too much time in the lab, but that's because it's fun. So with that, I would like to thank you as well.